Hello, and thank you for joining this presentation entitled Remapping Time Across Space. In this presentation, I'd like to chat with you about a paper that I've recently published with some co-authors in the Journal of Vision, and that article is entitled Remapping Time Across Space. It came out in 2013 with these co-authors. Some of you might recognize our recent graduate, Andrew Clement, from the class of 2013, and we also relied very heavily on some help from folks at Brown University, my doctoral advisor, Leslie Welch, and a former lab mate of mine, Elena Festa. Elena and I were both in Leslie's lab, and we were graduate students, and we both got our PhDs with Leslie. I decided to contact these two researchers at Brown University because of their expertise in visual attention, which I'd like to define for you now. Attention is one of these words that sometimes we in everyday life throw around maybe a little bit too loosely. And if you look at attention very scientifically, you'll find that there's a parallel between attention semantically and something like a word that's often used too generally also, and that word might be sleep. We can say that sleep is occurring when somebody is not responding to environmental stimuli, but if you make very careful and very precise physiological measures, you find that sleep is not this homogeneous state. In fact, sleep is electrophysiologically very heterogeneous. We have moments in which we're in stage 1, 2, 3, or 4, or REM sleep. The same thing is true for visual attention. We can say that in general, this is the selection of a sensory event, but we can break it down and say that there are actually different kinds of attention. And I'd like to give you a feel for two different kinds of attention. There might even be more than this, but for our purposes, we're going to speak of exogenous attention, or simply exo. And when we talk about exo attention, we're referring to a kind of attention that originates outside of the body, some kind of response to a stimulus, maybe a very loud stimulus that comes on all of a sudden, or a very bright stimulus, something that's very salient to us coming from the environment. We say that this then is bottom-up in our parlance. We can contrast this exogenous attention with what you might expect is called endogenous attention. So the counterpart terms here are going to be endo rather than exo. These are actually originating inside of the body. So something about the state of your brain is particularly important here in this endogenous attention. It's arising inside of your brain. This is said to be task-driven rather than stimulus-driven. So regardless of what the environment is doing, your behavior is under the control of some aspect of your body, maybe trying to pursue some kind of a task, and we call this top-down, in contrast to the bottom-up usage for exogenous attention. Okay, so maybe a picture will say 1,000 words. In this picture, we have a red team and a yellow team, and it looks like the red team is at the foul line trying to make a foul shot. So what we might remind ourselves of is that there can be a lot of stimulation in this room, and this player who's shooting the foul shot is going to use some attention to select a highly relevant stimulus to the exclusion of irrelevant distractors. And this player might be using something like endogenous attention, trying to select that and ignore all of this irrelevance out here. This would be task-driven. This player gets reinforced for making a point, regardless of what's going on in the environment and regardless of how the environment might be driving his nervous system's response to stimuli. Okay? By contrast, if we can imagine here that this player is shooting into a basket, and behind that basket we have opposing fans, they might be trying to create a distraction. What they really want to do is alter this player's behavior in such a way that the player is not able to make that basket. And so they're trying to disrupt this player's capacity to select that stimulus to the exclusion of others. So they start engaging in all kinds of hand waving, all kinds of noise making, anything to get the environment to change the state of the brain such that this is no longer steering the ship, the environment is steering the ship, the player misses the shot. Okay, we'll come back to that in just a moment after we understand a little bit about what might be happening in scenarios like this at the physiological level. And to do that, we're going to rely on some work that was done in recent years at MIT by Bushman and Miller. This paper was published in Science, and it involves some recordings that were made in the macaque monkey shown here. And I'd like to, for the moment, turn you into the macaque monkey and have you appreciate what this macaque had to do to earn a living during this experiment. So every day, the macaque monkey would be wheeled into the experimental room and would have to perform a task that either relies on what we would describe as exogenous attention, or exo, or endogenous attention, or endo. And the task went something like this. It begins with a simple fixation cross that is shown on the computer display, and then a sample stimulus is shown. it might not be clear from where you are, but the sample stimulus is a green line that's tilted a little bit anti-clockwise from vertical. Maybe you can see that right there. And then that sample stimulus goes away for some interval of time. After that sample stimulus has disappeared and the delay has elapsed, we come up here. We'll do the EXO trial first, shown on the top. That stimulus will now be placed randomly in one of the 4 quadrants around the fixation point, and the quadrant is varied randomly trial over trial. In this particular exogenous trial, here is the stimulus of interest in the upper left corner. What you might notice is that there are some distractors. And in this particular case, the distractors are identical to each other and very different from the targeted stimulus. The reason is these are purple or lavender in color, and they're very clockwise in their orientation. The target is unique. It's a very different kind of orientation from any of the other distractors, which are identical to each other in orientation. It's a very different color than the others. It's a very different wavelength. So even if you didn't remember this, even if you didn't have this stored in your memory endogenously, just by the fact that this is so different from the others and that these are so similar, that has to be the target. Not surprisingly, the animal makes a very fast response by way of an eye movement here, and the animal earns a juice reward. On to the next trial, and we're going to hypothesize here, or say hypothetically, rather, that we're going to get a very similar kind of start, but now we'll do an endogenous trial. So we get the sample, again, a green line, tilted anti-clockwise from vertical. Now we're in an endo trial, and you and I now know that that's the correct response, but we only know that if we can retain this endogenously in our memory. If we can somehow remember that that's what that was, then this would be the correct response. If we didn't remember that, we could look at the environment and say, I have a couple of anti-clockwise stimuli, but I have a couple of clockwise stimuli. I have multiple green stimuli, but I also have this blue stimulus. It wouldn't be entirely clear which one was the target because they're really all quite different from each other, unlike this case where the environment was basically handing you the correct response. Unsurprisingly, it takes humans and monkeys quite a bit longer to respond accurately on this exogenously defined trial. Okay? So what we can then say is, well, if the monkey is doing this, what would the electrophysiological data look like in the exogenous case or in the endogenous case? And to appreciate this point, I'll orient you by first telling you that here we've got the monkey facing this way. Here's the parietal lobe. I'll ask you to find that with me. You can go ahead and tap your parietal lobe. Here's the frontal lobe. And surgically, the researchers implanted electrodes and electrode arrays here in the parietal lobe and here in the frontal lobe. Okay? So what happened was when they looked at the data from exogenously defined trials, they tried to pick up on a particular dependent variable called neural synchrony. Sometimes when researchers like this are making measurements, they simply track the firing rate of a neuron. How many action potentials per unit time? And in many cases, that's a perfectly reasonable dependent variable. Here they're looking at a related yet distinct kind of dependent variable, and it's called neural synchrony. We're looking at many different neurons, say in the parietal lobe, and we're asking to what extent are they synchronizing their firing? Are they firing in rhythm together, so to speak? And they find something very interesting. When you've got an exogenous trial, then there is some level of synchrony that goes on, first in the parietal lobe, and then later into the frontal lobe. Okay? More than that, that synchrony was occurring in a relatively high temporal frequency range. That is to say, the temporal frequency range was something like 35 to 55 hertz. As these kinds of measurements go, that's a relatively fast rate. So to appreciate this point, I wonder if I can ask you to tap along or clap along with me. We're going to think about neural firing relative to some kind of rhythm, like maybe a quarter note rhythm. So if the quarter note rhythm were something like this, we'd need a relatively fast synchronized tapping rate, maybe something like eighth notes, which would be, okay, that would be the faster of the two rates. And that synchrony would emerge first here, and then would spread to over here. Inside of a classroom, what we might do is say that toward the back of the room, the more posterior portion of the room, just like the more posterior portion of the brain here, we would get the eighth note tapping, and then later on we'd see that eighth note tapping up here in the anterior portions of the brain. So I would clap like this and ask you in the back of the room to clap in eighth notes with me, and then people in the front of the room could do eighth notes as well, and we would have the spread from the posterior to the anterior shown here by this red line, indicating by these relatively small dashes that we have a relatively high temporal frequency spreading from the parietal lobe to the frontal lobe when we are engaged in these exo trials. Something very interesting happens when you change the trial type from exo to endo. and that's going to be shown here. Same diagram as before, but now we're measuring neural synchrony on endo trials, and we get the reverse sequence of cortical synchrony. That is, we get sequencing up here first, or synchrony here first, and then we get synchrony back this way. Okay? So in our example, we would get synchrony from the front of the room to the back of the room, and interestingly, it's happening at a much lower temporal frequency, which is about half the frequency that we had before. We might say this is an octave lower. We might know from music theory that an octave is a frequency doubling, so we cut the frequency in half, and we're playing quarter notes, not eighth notes this time. And these are originating, if you will, in the front of the room. The neurons start clapping together in quarter notes, and then finally that synchrony begins to make its way to the back of the room, the more posterior portion, and we have synchrony spreading in this particular case from here back this way. We have longer red lines to capture the idea that these are relatively lower temporal frequencies. Okay, so this is an idea about the electrophysiology, and we can remind ourselves then of how this might apply to an everyday scenario. As we've already said, here's our player, and the red team trying to make that foul shot and is trying to select that particular stimulus. That's the task, is to earn a point here and try to ignore the irrelevant distractors. The audience is trying to change this player's behavior, perhaps by altering the state of the nervous system, the state of the brain specifically. We can say that the player is trying very hard to engage in and to make use of the neural synchrony that might be happening first in the frontal lobe, and then going back to the parietal lobe. This is the endogenous variety, where we're trying to exclude the environmental distractors so that we can get our task done. And this person might be trying to tune into something like the 22 to 34 hertz range of neural synchrony. Meanwhile, the audience has different plans. The audience is thinking, I might be able to change that behavior if I can put that behavior under the control of different kinds of neural activity, maybe at a much higher temporal frequency, the eighth note pattern that we described a moment ago, and the pattern that is originating first in the parietal lobe and then moving to the frontal lobe. Okay, so this gives you some idea about what to think about perhaps when you're watching your next basketball game. What we can't do for ethical reasons is we're not able to put electrode arrays into human beings, but we can use EEGs, or electroencephalograph recordings. And in my own lab, we don't do this, but there are many labs around the world that do. And they perform, or they measure these electroencephalographic data while people are doing all different kinds of tasks. I'd like to show you some of the tasks that are relevant here, very similar to the ones that the monkey at MIT was doing just a moment ago. As always, we have some distractors, and in this particular case, we have black letter distractors. These 20 letters from the English alphabet make up the distractor set, and all of them are going to be present on every one of the trials. On any given trial, we might pick one target, maybe from this set. That set is D, F, G, J, K, L. These 6 red letters are chosen from the center row, or the second row, I should say, of a computer keyboard. And you might notice that these are said to be exo targets because any one of them will differ from the background physically. These are red, that's long wavelength. These are broader band, but they're very low in luminance. So a light meter can tell us that this is distinct from that. That's a physical difference. We would call that an exogenous difference. By contrast, we can, on other trials, scramble up these one more time and pick a target, but we might pick it from this endogenous set. It's consisting of these numbers, 1, 2, 3, 4, 5, 6. Okay? All of them are black, just like these are black. Now you might object and say, well, clearly these are numbers, those are letters. Aren't they different categories? On some level, yes, they are different categories, but that category is endogenously defined. And what I mean by that is that you and I have learned, and in our nervous systems, in our brains, we know that these are letters and these are numbers and these are letters. But what we can say is that you could go to some member of a different society who hasn't been exposed to this, and they wouldn't know that this corresponds to the numerosity 4. They wouldn't know that this corresponds to the phoneme T. For all they know, this could be a T sound, and this could be a symbol that represents the numerosity 4. These associations are entirely arbitrary, and they become meaningful only to those folks who have, in their brains, in their nervous systems, made these associations. Okay, so this is an endogenous association. This is an exogenous association. So what we'll do is we'll give you a couple of visual search task trials, and we'll ask you to engage in target identification. This is one of 3 possible tasks that we could have you do, and we'll develop the others as we go along. In the target identification task, I'm going to scramble up all of those 20 black letters, and I'll pick either one of the red letters or maybe one of the black digits, and I'll ask you to call out that response or call out the target as quickly as you can find it. Okay? So I'm going to flip the screen in just a moment here and call out the target when you see it. Here we go. Okay? So hopefully you can see that. I might ask you to think about whether this is an exogenously defined trial or an endogenously defined trial. You might think about that. And we'll go on to our next trial. We're going to, again, engage in target identification, one of 3 possible tasks that we could have. I'll ask you one more time to call out the target when you see it. I'm going to scramble up the distractor set, pick one particular target, and I'll ask you to call that out when you see it. Here we go. Okay. Hopefully you can find the target in that. And we would ask you one more time, do you think this was an endogenous or an exogenously defined trial? Okay? So that gives you the idea about what would go on in a visual search, which is something that we can do to explore the spatial resolution of attention. So for example, back here on this display, I can manipulate very systematically the interletter spacing, sometimes making that spacing very small in space or in distance, sometimes greater. I can have larger or smaller letters. I can manipulate that to find out just how finely your visual system can respond to select a particular target to the exclusion of distractors, the spatial resolution of attention. I can play the game a little bit differently and ask about the temporal resolution of attention. I can ask how fine a unit of time is your attentional system able to reliably distinguish. And to do this, we introduce a new tool similar to the other one, but a little bit different. It's called the Rapid Serial Visual Presentation, or RSVP display. And in just a moment, I'll show you a schematic of this RSVP. And on every trial, we show you one of these dynamic sequences, and we ask you to respond, s'il vous plaît, okay? RSVP. Here's a schematic of what would happen on an actual trial. We can keep exactly the same black letter distractors that we had a moment ago. We can keep exactly the same red exogenous targets and black digit endogenous targets. We can keep all that the same. You can see that we get different frames of a movie. And what's interesting here is that these stimuli on any given moment can only appear in one of 2 locations, just to the left and just to the right of a central fixation cross. So unlike the other task where there was a lot of spatial uncertainty, here there is no spatial uncertainty. We know that the targets and the distractors are going to land frame after frame right in the same location. What we don't know is when they're going to be appearing. And they're only flash very briefly. In this particular case, hopefully you can see that if you were to engage in a letter identification or target identification, you would say D and 3 are the targets in this particular case. We can ask other kinds of questions, maybe about the relative timing. And here are the 2 other tasks that I alluded to earlier. One is called a temporal order judgment task, or a TOJ task. And here, you are not obligated to identify which letter that was or which number that was. Rather, you simply have to say that either the letter came before the number or vice versa. You would say letter first or number first. Those are your 2 possible response options on this 2 alternative forced choice or 2 AFC test. We call that a temporal order judgment. It tells us about relative timing. The other task that we have that tells us about relative timing is called a simultaneity judgment task, or an SJ task. Unlike the TOJs, temporal order judgments, these are simultaneity judgments, or SJs. And here your 2 response options are same and different with respect to these 2 targets in time. You would report that either these 2 targets were flashed at exactly the same time, or they were flashed at different times, without having to tell us anything about temporal order, certainly not telling us anything about the particular letter or the particular number, just same or different kind of judgment. Okay? All right, so you might say, why would we care about this temporal resolution of attention? And there are 2 answers to that. One pertains to basic research, one pertains to applied research. I am predominantly a basic researcher. I want to understand how the visual system works. It might be the case that down the road, several years from now, a good description of how the visual system works will help solve some applied problem, but right now I just want to know, basically for the sake of knowledge. I'm intrigued by the rate at which you can extract information from the environment, and just knowing that is something that I find satisfying. Other people prefer to have an applied response to this question about who cares regarding the temporal resolution of attention. So lots of ways to go here. A simple example would be traffic safety. And what I can share with you is that lots of folks have been measuring over the years the temporal resolution of attention. And a ballpark figure that you might have in the back of your pocket after you finish watching this presentation would be a 10 Hertz sampling rate. That is to say that at 10 Hertz, we're picking up a sample from the environment once every 100 milliseconds, or 10 Hertz. Now, with regard to traffic safety, we can suppose that you're driving in a car at 70 miles an hour. If you're driving at 70 miles an hour and you're sampling at 10 Hertz, or once every 100 milliseconds, that's the equivalent of taking just one sample from the space around you every 10 feet. That's a relatively coarse sampling mechanism. Okay? So this could be a 10-foot difference, would be a life versus death difference in a crash. So really the temporal frequency of your visual attention matters very much, particularly as you're moving at these fast speeds. You can imagine an even coarser sampling of space if you're flying an airplane that's moving at much faster than 70 miles per hour. The National Traffic Safety Board offers us this little reminder about the physics of the situation. Crash energy, okay, so when the two objects are colliding, is proportional to the square of impact speed. Okay? So just a small change in impact speed has a relatively large effect on crash energy. So that has very, very clear implications for how badly you're going to be injured, how much property damage you're going to do. In short, milliseconds matter because of this squaring relationship. So this is one of the reasons why we might say that the temporal resolution of attention actually is very important, even in everyday life, and this is why we say that RSVP experiments are to the rescue. So let's take a look at some RSVP experiments. Some of these were done by a research group led by Rolf Verleger in Germany. And the Verleger group published in 2011 and 2012 articles in the Journal of Psychophysiology and the Public Library of Science, where they've made some electroencephalographic recordings similar to what we had seen earlier. And the participants are now performing not a visual search task like the monkeys were doing a while ago, but an RSVP task. Okay? So what they found was something quite interesting. They found something interesting behaviorally and something very interesting electrophysiologically that I'd like to share with you now. First, the behavioral component, there was greater accuracy for targets in the left visual field than for targets in the right visual field. So here's our temporal order judgment sequence, our RSVP sequence rather, and we have a maybe red letter L over here, and then we have the numerosity 4 happening in your right visual field. Okay? Even if these are landing on your retina at exactly the same time, they don't appear to be getting all the way back here to the cortex at the same time. It appears to be the case that we're getting a hastened response to the left visual field stimulus than we are to the right visual field stimulus. We're also more accurate. We have greater accuracy in the left visual field, and we're getting a faster neurological response to that left visual field stimulus, maybe on the order of a few tens of milliseconds, maybe almost 100 milliseconds. Okay? So this is quite interesting. Also, it was quite distinct with respect to the location of the brain where this hastened neural response had been observed. Strictly speaking, we call this the N2 PC component of the EEG data. The N2 stands for the second negative going spike in the EEG trace, and the PC stands for parietal contralateral. So to put that all together, what we're going to say is, here are the two stimuli that come onto the screen. This is an L, this is a 4. This L is on your left. We actually get in the contralateral cortex, which would be your right cortex, we get a pop in the electroencephalographic data sooner in your right parietal cortex than we get in your left parietal cortex. So the N2PC, which is known to be as a marker for visual attention, is showing this kind of physiological hastening. Very interesting and really quite surprising. And in this particular case, the participants were performing a letter identification task better on the left than in the right. The fact that you have this physiological hastening, an asymmetry in the neurological response, raises for me very important perceptual questions. Given the hastened left visual field neural response, so left visual field right parietal lobe, is it the case that LVF targets are perceptually available sooner than RVF targets? Okay, so these two targets are physically synchronized out here in the real world, but given that the response neurologically to one of those is hastened, is that hastened neurological response perceptually available to you? It's an interesting question. It is not a foregone conclusion that every event inside of the brain has a perceptual correlate. There's perhaps far more neural activity than your attention can select at any given moment. Your attention has to be selective and pick out one type of neural activity over another type of neural activity. So it could be that we get a hastening, but we actually don't have a perceptual correlate of that. Or maybe there is a perceptual correlate. That's the first question that we're going to ask. Then we can ask this question. Again, given the hastened LVF neural response that Verleger's group had empirically observed, how does the visual system estimate simultaneity? One more time, if these are the two stimuli and they come on at the same time, we'd like to be able to make an accurate judgment about that. And yet it might be the case that if our visual system is introducing an asymmetry, maybe we've lost the capacity to estimate simultaneity particularly accurately. So what we're going to do is engage in a couple of experimental tasks in what I'll show you next. These are two tasks. These are counterbalanced within subjects, and we have Denison students performing these tasks, and we have a total of 23 such students. One of the tasks I alluded to a moment ago is our TOJ task. This is the temporal order judgment task. Which target came first? Either that exogenous red letter, shown here, or that black number, the endogenous target, shown here. Okay? So which of those two targets was coming first? That would be the temporal order judgment. On other blocks of trials, again, randomly counterbalanced within subjects, we have simultaneity judgments, or SJs. And this is too about relative timing. But what we can say is that the target timing question here is, were the two targets shown at the same time, or were they asynchronized in time? Maybe by the letter leading a little bit or leading a lot, letter lagging a little bit, letter lagging a lot. Did they happen at the same time or at different times? Across these two tasks, we've very importantly kept retinal stimulation exactly the same. So the bottom-up feed is identical here and here, and we're wondering if there are any task-related differences. The independent variables are ones that we've largely inherited from Verleger's group so that we could extend their work. The first type is the target type. We have either exo, those would be the red letters, or endo, those would be the black digits. And as always, we use the black letter distractors that you had seen before. Verleger had used those too. We get target asynchronies. Sometimes it's the case that the letter is very much preceding the digit target. Sometimes they're synchronized. Sometimes it's lagging behind. Minus 700 to positive 700 is the range of asynchronies that we've been entertaining. The most important two variables are shown here. One is hemifield. It has two levels, the left visual field, LVF, and the right visual field. So across trials, we're going to have some letters on the left, numbers on the right. Other trials will have numbers on the right, letters on the left, and so forth. Those are entirely random trial over trial. What we're going to do is use a little bit of symbol here where we can have the left visual field targets when we want to refer to them. We'll put those in blue for this presentation, and your mnemonic to help you with that is to think that the letter L is shown here and it's in blue, and there's an L in blue, and here we have right on red. Hopefully that would help. If you want a second mnemonic as a lifeline in the busy graphs that are about to come up, you can think of the American political spectrum and the map of the United States politically on the evening of an election. The politically conservative right-leaning individuals are typically shown in red, and the left-leaning liberals are typically shown in blue. So hopefully that's enough mnemonic to work for you, left versus right hemifield, and then we have these two tasks, TOJs versus SJs. So that's a fair number of independent variables. To make things a little bit more manageable, I'll show you these data first for TOJ, and then I'll show you the same format for SJ, and we'll see what we get when we're all done. Our dependent variables are going to be essentially and conceptually perceived relative timing. The way that we operationalize that on the TOJ task is we can take one of our two response options, for example, the letter first option, and we can just track that and ask what proportion of trials are the participants responding with the letter first response. Okay? You can think of it this way. We really don't need to track both of them. If we were flipping a coin 100 times and we got 75 heads, we could know from that that we had 25 tails. We only need to track one of the two. There are only two possible responses in each of these two alternative forced choice tasks. So likewise, on the simultaneity judgment task, we only need to track one of the two possible response options, and we'll track the different responses. Okay? So here we are with a plot of the data in just a moment, just to lay it all out so it's not too confusing for you. On this axis, we have the target asynchrony. In the center of this x-axis, we have physical synchrony, and over here we have the case where the black number is coming first. Over here we have the case where the red letter is coming first, and they're asynchronized by various amounts. You can think of it this way. We'll make this a mantra to help you understand these. Here's letter is lagging. On this side we have letter is leading. Can you say that with me? Letter lagging, letter leading. Okay, and that's the physical axis. On this axis, we have the proportion of letter first trials because this is the TOJ task that we're looking at now. We call what I'm about to show you a psychometric function because, as I've shared with my friends in physics before, what we typically track in my lab is a psychophysical function, and we have this little gesture that we make. Psychophysics. I'm a psychophysicist, and I'm always plotting the psychological variable on the y-axis and the physical variable on the x-axis. Okay? So let's go ahead and now show you some data and see if we can understand how to unpack these data. First, a couple of similarities between the blue data series and the red data series. One obvious similarity is that in both cases, the participants' behavior is very much under stimulus control. And what we mean by that is that the proportion of letter first responses is nice and high when in fact the letter is leading by quite a large margin. Conversely, when the letter is lagging by quite a large margin, participants very rarely say that the letter came first. So the participants' behavior is very much under stimulus control. What might strike you as very salient here is the leftward shift of the blue data series and the comparative rightward shift of the red data series. And there's a particular point that I want to call your attention to. We call this point the point of subjective equality. In our psychophysics, we can say that the point of physical simultaneity is when there's no asynchrony. We're here at the 0 point. With regard to the subjective simultaneity, we can say all the way up here, the people are always responding letter first. Down here, they're always responding number first. Here, they're completely on the fence. This is the halfway mark on the psychological axis, just like this is the halfway mark on the physics or physical axis. And what we can do in psychophysics is try to find the association between the subjective experience and the physical stimulation. And what we're going to carry out is a simple computation where we start at 0.5, we move on over until we get an intersection, and then we drop straight down, and we find out that this blue line lands at the minus 83 millisecond mark. That's shy of physical synchrony. It's 83 milliseconds sooner than physical synchrony. If we play the same game over here for the red data series, we go a little further off to the right, and then we drop down, and that's a positive 51 milliseconds. So this is really quite interesting. Just by changing the position of the letter, and here's the letter on your left, here's the number on your right. When we show you this kind of a sequence, we tend to get leftward shifting, We tend to get leftward shifting, okay, shown here in blue. All I have to do is reposition the targets into this configuration. I'm changing the space, and I'm altering the perceived time. The psychological time now shifts very much rightward just by changing the space. So it's an interesting correspondence between spatial position and perceived time. I wonder if I could point out a couple of other components here in this graph. We can take, for example, physical synchrony, when the two targets are physically on the screen at the same time. If the letter is in the left visual field, look how frequently it's perceived as coming first. We move that same letter over to the right visual field, and we now reduce the proportion of letter first responses by many, many standard errors. A very, very large effect. Another way of saying the same thing is by looking at what's going on here in these gray ovals. You can see that we get a pretty good match in the blue and red data shown here. But in order for the red to match the blue, the red has to be delayed by something like 134 milliseconds. Here, here, and here. As we move out to the extremes, that gap tends to close. But when the transition is very, very subtle, and when the difference in timing is very, very subtle, we do see that it appears to be the case that we have a hastened response to the left visual field stimuli. That's shown much more simply in this graph, where I'm plotting the point of subjective equality for the case in which the exotarget is in the left visual field, shown here in blue, or in the right visual field, shown here in red. And you can see that they're pointing in opposite directions. Each one is comfortably away from physical neutrality, and there's actually a 134 millisecond difference between those two. Okay? So the answer to question number 1, and just to remind you, that was given the hastened LVF neural response that Verlager had recorded, are LVF targets perceptually available sooner than RVF targets? Okay? And the answer to that overwhelmingly is yes, and the difference was something like 134 milliseconds. So we can pause for a moment here and try to put that 134 milliseconds into some kind of context. And you might recall from a few slides ago that we said that the temporal resolution of attention is maybe on the order of 10 hertz, or having temporal periods of 100 milliseconds. It appears to be the case that something is available to you in your left visual field about 134 milliseconds sooner than it would be were it in the right visual field. That is approximately a full attentional cycle. So it's almost as if our left and right cortices are out of whack by maybe as much as one attentional cycle. An absolutely stellar observation, I think, really quite interesting. And we got the lead on this because of Verleger's good EEG work. It appears to be there psychophysically as well. What we can try to do then is try to explain this by maybe understanding an algorithm. And this wouldn't have to be the way that the visual system is working, but we might use this as a working description. In this algorithm, what we're going to do is take in some inputs and try to come out with a judgment on a TOJ task. We'll call this the TOJ algorithm. And remember that we might say something like letter first or number first, or that the left visual field came first or the right visual field came first. So how do we get to one of those two conclusions? What this algorithm poses is that the visual system regularly registers time of arrival. It does so in the left visual field. It does so in the right visual field. And for any given target that's coming into the cortex, we can get an estimated time of arrival, one for the left visual field target and one for the right visual field target. And we can assign simple millisecond values to each of those. In a hypothetical example, we'll say that the left visual field comes in at millisecond number 1. This one over in the right visual field comes in at millisecond number 25. So we can do 1 minus 25, we get a negative 24. If we forget about the bias just for a moment, we'll turn that down to 0. That negative 24 is less than 0, it's a negative number. We would say left first. If the sequence had been reversed, if it were the case that this came in first at the 1 millisecond mark, this came in at the 25 millisecond mark, we would do 25 minus 1, we would get positive 24. Assuming no bias, that would be a positive number, and we get the right visual field occurring first. What we can say is that these components are entirely stimulus-driven. Stimuli are landing on your retina at various times in these RSVP displays, and the visual cortex is presumably registering at least roughly what those times of arrival would be. The bias is really quite interesting. The bias is a stimulus-independent top-down factor. And one way of thinking about this is that it might be akin to the spread that one would have to beat in a gamble. So for example, let's presume that the bias is also measured in milliseconds. And those can be negatively or positively valenced milliseconds. We are talking about time after all. If this bias is something on the order of positive 100 milliseconds, then a difference of either 24 milliseconds here or negative 24 milliseconds gets swamped by this positive 100 milliseconds, and we wind up with some value that's greater than that is greater than 0. And when that's greater than 0, we're going to say right first. If we had a negative 100 millisecond bias, that would be a negative number, we would say left first. So depending on how big the bias is, that might actually overrule what's going on in the stimuli. Okay? So you can think of having two different teams, and we can either compare them straight up, or we can have them compared after we say that one team is given 7.5 points as a hedge of some variety. So simply by adding some of these top-down effects and these stimulus-driven effects, we might be able to offer just a crude way of thinking about how TOJs might be performed algorithmically. Okay? So with that as some context, why don't we see if we can go on to question number 2. And just to remind you, we can say that given that the LVF targets are perceptually available sooner than RVF targets, okay, that's what we found in experiment 1, how does this visual system estimate simultaneity? That's an interesting question. Okay? All right, so there are different kinds of hypotheses. We'll start out with the simplest one. Okay? Our most parsimonious hypothesis would be that TOJs and SJs rely on the same neural events. So we could use a scheme similar to the algorithm we saw just a moment ago. We could get a left visual field, estimated time of arrival, one for the right visual field. We'll combine those. Maybe we have a bias, maybe we don't. We could add that in, and we do one thing if that number is negative. We make the alternative response if that number is positive. Really, same kind of a logical scenario. And our mantra might be something like this. What works for TOJ works for SJ. This would be very parsimonious because we could use one algorithm that would explain these two different tasks that are relative timing tasks. And we do love parsimony as scientists. And later in the discussion section, we might chat about why it is that we love parsimony. But this would be a very parsimonious way of making some predictions about the relationship between TOJs and SJs on these rapid serial visual presentation tasks. What I'd like to do is now show you the data from the simultaneity task And on this y-axis, we're going to be plotting, as we said a moment ago, the proportion of different responses. So the people are making same different judgments. We'll track one of those two responses, proportion of different responses. The x-axis remains exactly the same. In fact, retinal stimulation is exactly the same here, which is to say we're going from letter lags through physical equality to letter leads. Okay? Now, if we think that our hypothesis is correct, a very interesting prediction occurs. And that prediction is shown here. Remember that a moment ago we had described that there was a leftward shift in the point of subjective equality for this blue symbol data series. That's the case where the exogenous target happened to have been on the left, kind of like this, with the black digit target on the right. The blue data series is sequenced that way or symbolized that way. The red data series has just the opposite spatial configuration, and we had seen left and rightward shifts. So if it were the case that the point of subjective equality on the TOJ task were to be rearing its head here on the SJ task, then we would make the prediction that if you're plotting the proportion of different responses, you would see a lot of different responses when you have a gross asynchrony on this side, very many when you have a gross asynchrony on this side. As you approach physical neutrality, we should get to something like a minima in that curve, but the minima probably won't be at 0 if our hypothesis is true. That is, if what's good for TOJ is working also for SJ, we would expect a leftward shift in our minima for the blue data series, and correspondingly perhaps a rightward shift for the minima of our red data series. Well, here are the data, and what I can tell you is that this prediction is actually wrong. Very much so. Let's take a look at why it's wrong. We'll follow the blue data points. The prediction was that the lowest of the blue data points should be somewhere left of center. In fact, it's really very much dead center, and it's quite a few standard errors from whatever the next closest one is. The same thing is true for the red data series. We might have imagined that there could have been a rightward shifting point of subjective equality, point where these are least different and most similar, but instead we get now a several standard error difference between this datum point, this datum, and those two. And so the minima is occurring for both the red and blue data series right at physical equality, unlike what we observed just a moment ago. So we're getting wildly different patterns here. Our left visual field hastening seems to have gone away, even though the retinal stimulation has remained the same. So what we can say then is regarding question 2, and that question was, how does the visual system estimate simultaneity? And do TOJs and SJs rely on the same neural events? And these were the neural events that we had invoked. It turns out no. It is not the case that we can use exactly these same components to come anywhere near explaining the data set that we empirically observe. Okay? So then we might say this by way of summary. TOJs exhibit this left visual field hastening, but SJs did not do so. And this tells us that the TOJ and SJ tasks seem to be obeying distinct rules with regard to the underlying neurology. We're only performing behavioral experiments here, but it really can't be the case that we had a shared neural response on these two different kinds of tasks. There must have been something unique about the task to drive that very, very salient difference that we observe. Okay? The TOJ and SJ results differed despite identical retinal stimulation across tasks. So it's interesting to muse about that. We held very deliberately the retinal stimulation identical on the TOJ and SJ tasks, and yet we get these very strong left visual field leading biases on the TOJ task, and really that is wiped away on the SJ task. Okay? So where did the LVF hastening go on the SJ task, you might ask? And we'll have to come back to some kind of new algorithm that might be similar but can't be identical to the other one because the data would not support identicality, no matter how parsimonious that might have been. So we're going to invoke now a two-step algorithm where we begin by simply adding a delay to the left visual field estimated time of arrival. Earlier we had seen that the left visual field had been leading, but if it were leading on this SJ task, we also would have expected a minima left of center, and we didn't see that. So the visual system somehow, at some point, must be adding a delay of some variety, remapping essentially the left visual field estimated time of arrival. And then our second component is similar to what we had a moment ago. We can take the absolute value of the newly adjusted, the remapped LVF ETA. We can now difference that with the RVF ETA, and we take the absolute value of that. If that's greater than the bias, remember this is the spread that we would have to beat in a gamble, then we would say that these two are sufficiently different. If the absolute value of this difference is somewhat close to 0, then we're inclined to say same. Okay? So this little step constitutes the remapping in the title of this talk. It appears to be the case that we do get a left visual field estimated time of arrival remapping. This is a remapping of a bottom-up signal that was registered in our cortex. The hastened neural response is now remapped to a later time in the RSVP sequence. That would be the thinking that goes on here. Okay? So you might recall that we had this very elegant and parsimonious hypothesis that the TOJs and SJs relied on the same neural events. And we had this mantra, what works for TOJ works for SJs. Unfortunately, that is not correct. We do like, in general, parsimonious hypotheses. These are very elegant. But as a wise person once said, elegance is for tailors. So why don't we make that our second to last mantra? If you'll say it with me, elegance is for tailors. Our final mantra is the universe doesn't owe us elegance. Everybody, the universe doesn't owe us elegance. And then finally, we won't make it a mantra, but we'll look at one more great thinker of our time. There we are. So thank you for attending to our talk.